Okay, so today's notes are on antibiotics. So there's going to be this PowerPoint about the notes, and then after that we're going to have another video on labeling the diagram of how antibiotics work. All right, so let's just get started. All right, before antibiotics we had very little to try to treat infections. Ancient people used things like mold or soils or plants or herbs to try to treat infections. There's ancient scripts from Serbia and China and Greece, which shows using moldy bread on wounds to try to cure the infection. And it kind of makes sense when you figure moldy bread has bread mold on it, and a lot of the antibiotics that we have originally come from molds or fungus. The ancient Nubians used, or they brewed beer, and what paleontologists have found is that when they look at the bones, they found that there's a lot of tetracycline in the bones of some of the people from this culture. So what they figure is that the Nubians actually knew they were culturing or preparing beer in a way that helped to fight infection. They just didn't know they were making tetracycline. All right. We also had our patent medicines, which contained a lot of opium and alcohol. And remember, patent medicines didn't actually treat the illness. They just treated the person, made them feel better about their illness, right? Basically made them high or drunk, so they didn't feel how sick they actually were. In 1910, Ehrlich, who was a Jewish German scientist, he found a chemical that he could use to treat syphilis. It wasn't an antibiotic, but it was actually a actual chemical that could, it was an arsenic derivative that could treat syphilis. We also had phage therapy that was used in Paris around 1910 that was used to treat dysentery. So remember, that was using the bacteriophages to kill the bacteria that were causing the dysentery. There's a whole pile of other kind of concoctions that people use to try to fight infections. But it was in 1828 that Fleming is given the credit of discovering penicillin. All right, so here is Fleming. All right, in his story, he was a Scottish researcher that was working in England at St. Mary's Hospital. All right, so the story goes that in the summertime he went on holiday and just kind of left his lab. Now he was growing Staphylococcus aureus in his petri dishes because Staphylococcus in that time used to cause a lot of infections. It's just a bacteria that lives on your skin, happily lives on your skin, but if there's an opening and the Staphylococcus gets in there, it can cause a lot of infection, a lot of serious infection. All right, people used to die of like the littlest, like a razor nick. You could die of something like that. So he went away on holiday, came back, and then he was going through his plates, taking a look at them, and he noticed on one of the plates that there was this growing of mold, and around the mold there was this zone of inhibition. Right, so the next slide will show that. So here we see, here's the mold that's growing. This is his picture. And then here is the zone of inhibition. So here's the Staphylococcus aureus growing here. And then around here are either they're dying, they're undergoing lysis, as this says here, or there is no Staphylococcus growing in here. So he knew that there was some type of substance that was being exuded by the penicillin mold. And he tried to extract it, but at the time the technology wasn't very good, so he just kind of put it to the side, wrote a paper about it, and then put it to the side and really didn't do very much more with it after that. Now, even before him, people had discovered that, again, there were some of these penicillin molds that could kill bacteria, and again, they couldn't isolate it. In fact, in 1897, there was a French scientist whose last name was Duchenne, and he did a thesis as part of his research that guinea pigs treated with this substance that was secreted by penicillin mold would recover if they were infected with some type of bacterial infection, and he found that those sick guinea pig actually did recover. His thesis wasn't recognized by anybody who was important, and then he ended up entering the French army, and he just kind of stopped his research. So because Fleming couldn't isolate this substance that was out here, he kind of put the research aside as well. And about 10 years. So these next two guys, Fleury and Chain in 1939, they managed to isolate penicillin. Now Fleury was an Australian who was living in England, and Chain was a Jewish German who was living in England. His real name is Ernst. I think they've Americanized his name. He was in Germany, and he left in 1933 because he didn't like Hitler's stance on Jews. So he escaped. Well, he didn't escape, he just left, but his mom and his sister ended up being killed in the concentration camps. So they isolated enough penicillin in 1939 to be able to do some experiments. So what they did is they took 8 mice, they gave them all this Staphylococcus or Streptococcus bacteria, and they were all very, very sick. So the 4 mice that they gave the penicillin to survived, and then the 4 mice that they didn't give the penicillin to, they ended up dying. So then they were growing penicillin in basically whatever they could find, whatever vessel that they could find around the labs in England. Now, England was in war at this time, so they didn't have very good luck getting around and going to industry and saying, can you build some chambers so we can grow this penicillin mold? Because in England, remember, they were being bombed and attacked. So they had to just basically grow it in whatever they could grow it in, and then eventually they ended up going to the United States and getting United States laboratories to grow the penicillin for them. But anyway, then they isolated some more penicillin, and the first human that they tested this on was a policeman who actually got pricked, I think in the cheek, by a rose thorn, and it ended up getting really, really infected. It ended up getting sepsis and getting into his bloodstream, and they were treating him. Now at this point they didn't know how much penicillin to give him and how long to treat him with penicillin, and he started to get way, way, way better, and then they ran out of penicillin, and then the police officer ended up dying. So they vowed never again to be able to do an experiment unless they knew they were going to have enough penicillin to get through the whole entire treatment. So they went to the United States and they got some help to grow the penicillin, or to grow the penicillin mold in the United States. Now what was really neat is different strains of mold will grow more or less penicillin depending on the strain, and they ended up finding a strain in Peoria, Illinois, which is where they were doing the research, that they found it on a moldy cantaloupe in a market in Peoria, Illinois, and this strain of penicillin produced a ton, or penicillin mold produced a ton of penicillin. So basically they stayed in the United States for quite a few years and did some research on this penicillin mold and penicillin. And the Fleury guy, he wanted to patent it, but Chain said, no, this is something that's good for all mankind. So the result of all that in their research was that they ended up being able to win the Nobel Prize in 1945. So this is Fleming, this is Fleury, and this is Chain, and they are credited with saving more lives than all the Nobel Prize winners put together. So that was kind of nice that all 3 of them got it. Now he would speak quite publicly about his discovery, and he worked with these 2 guys, kind of, they all kind of worked together. And then these 2 guys also had a lot of scientists working with him as well. But these 2 guys almost didn't get the Nobel Prize because people just thought that Fleming, because he just loved to talk. Remember, he's Scottish, so he's going to be out there with the gift of the gab. So he got a lot of credit for it at first, but then he spoke out and made sure that the Nobel Committee knew that these other 2 guys were important here. Okay, so let's do, and there's so much more history with all this kind of stuff. Again, I could go on for days, but we only have one lesson to get through. So antibiotics work in a couple of ways, all right? So they can either be bactericidal, so the cidal part meaning killing, so this is going to outright kill the bacteria, or we can be bacteriostatic, so where we kind of just keep things the same, so where we prevent the growth or the reproduction of the bacteria. All right, now some antibiotics can actually do both of them, and that depends on the dose of the antibiotic, it depends on the duration of treatment with that antibiotic, and it depends on the state of the bacteria. All right, so here we have again, this is just a cute picture to show you bactericidal. Now that was spelled wrong on the previous page. It says bacteriocidal. It should say bactericidal. I just don't want to have to redo the whole slide thing again. So bactericidal basically just kills the bacteria right out. Here's some antibiotics that you may be familiar with. Now, I'm not going to ask you any of the names of specific bacteria. You just need to know they're bactericidal or they're bacteriostatic, all right? It's just kind of interesting having these names because if you ever are prescribed antibiotics, you'll kind of have a little knowledge of whether you're outright killing the bacteria or if you're just preventing them from growing properly. All right, so here's some of them. You've probably been on a sulfa drug before, or if you're doing tetracycline for acne, or erythromycin if you've ever had an infection, they'll give you some of these things. So these ones just kind of like prevent them from growing any, like reproducing. Okay, so how antibiotics work, and this is what the next lesson is going to be on the mechanics of antibiotics, but this is basically what they do, all right? So they inhibit cell wall synthesis or function, because remember the antibiotics have a cell wall. Our cells don't have a cell wall, so we can use antibiotics go in there, ruin the cell wall, and it doesn't touch our cells. All right, we can disrupt our cell membrane. Now we have cell membranes at all, but the antibiotics are going to target certain types of phospholipids that bacteria cells have that human cells don't have. All right, we can inhibit folic acid synthesis. Again, bacteria can make their own folic acid. We can't, we have to get it from our food. So we can inhibit bacteria from making folic acid. We can inhibit them from reproducing. We can also inhibit nucleic acid, so either DNA or RNA synthesis or its function, and you'll see how the antibiotics do this a little bit differently in bacteria cells than they do in human cells. Or we can inhibit protein synthesis, and you'll see how this is a little bit different here in bacteria cells. Okay, so that's that for this little lesson, and the next one will be on labeling a diagram of how different types of antibiotics work.